Okay, the next thing I want to talk about is something similar to a monohybrid cross. It's called a test cross. It's basically using this technique to solve a problem. So let me give you a hypothetical example. You've got two flowers here. They're from the same species. Flower colour is controlled by one gene, and it exists in two alleles. So the capital P is the dominant one, and that's purple. And the recessive one, lowercase p, gives white flowers. Now, if you were to go along and find an individual that looked like this, purple flower, it could be homozygous dominant. It could be two capital P's, but it could also be heterozygous, one capital P, lowercase p. You don't know. A test cross lets you figure out which it's going to be, and it does it always in the same way. In order to understand, to try and find if you've got this allele hiding in there, you have to cross this flower here with a homozygous recessive. If you cross this flower with a recessive, then anything that is recessive and hiding in the genotype of the purple flower will be expressed. So it always works the same with the test cross. You breed it with a homozygous recessive. So I've done the two crosses down here for you. These are our purple flowers here. These are the two possible outcomes, okay, that a purple flower could have. And here, these are my homozygous recessives. These are white flowers. So I've shown you the potential outcomes here. I've done it already. If you get a heterozygote, then 50% of the offspring will be purple. If, however, it is a homozygous dominant purple flower, then 100% of the offspring will be purple. And the proportions that you find after breeding will tell you which is which. Okay, now this is the worded example I want you guys to do, so you can pause the video in a second. It says red hair is caused by a recessive allele. Show how a test cross could be used to determine the genotype of a non-red-haired person. I had to add that little typo, but non-red-haired person. So you find a non-red-haired person, what are you going to do to work out their genotype? Okay, breeding experiments on humans typically frowned upon, but just imagine and have a go at this one. All right. Okay, let's have a little look at the worked example of this one and check that we're all in agreement. So we have a non-red-haired person. Now, given that this is the dominant one and that's the recessive allele, we know that they are going to need at least one of those alleles there, but they could be heterozygote or they could be homozygote. We want to find out what their genotype is, so we are going to have to breed them with a red-haired person. So here are the Punnett squares for that, and you'll get... And so it's exactly the same as with the flowers. You're going to get 100% of the offspring are going to be non-red hair. Okay. But over here, you are going to get 50% of them are going to have red hair, and 50% of them are going to have brown hair, or non-red, or whatever we're calling it. Okay, so that is what's called a test cross there. Next example, also using the monohybrid inheritance ideas, is something called codominance. Up until now, we've been looking at situations where when you put two alleles together, one can completely dominate the other. So this flower here would be every bit as purple as that one there. This one completely dominates. But it's not always the case. Sometimes you can end up with two alleles that both influence the phenotype. So the example I've given here is flowers again, and we've got two codominant alleles. Capital R gives red coloration, lowercase r gives white coloration, but heterozygotes, instead of being red, which is what you'd normally get, in this instance you get an intermediate phenotype of pink. You use a genetic diagram to work out the phenotypes of the offspring of two pink flowers. Okay, they've told us they're pink, which means they've got to be heterozygous. So that means our two pink flowers are going to be that genotype. So that's our cross right there. Okay, so these are the two parents. We've got heterozygote breeding with a heterozygote. Now what we've got to do is work out the gametes, where we're going to undergo meiosis, end up with some haploid cells. It's going to look like that. It's going to look like that. There is my Punnett square. So here are the genotypes. Okay, let's have a little look at what we've got here. So this one, we know that that one is going to be red. This one we know is going to be white, but it's these ones here, they are both going to be pink. Now if you think about the ratio, we've got 1 to 2 to 1, so we would go red to pink to white, 1 to 2 to 1. Okay. Now that's my made-up example. This is a real-life one. Blood types. It's determined by 3 alleles, A and B. They're the codominant ones. And then there's a third allele in there called O, which is recessive. So I want you to show how parents with blood type A and B can produce offspring with 4 different blood types. So have a think about that one. We pause the video, have a go. Okay, let's see how we got on with this. So let's have a look at the parents. Let's work out their genotype first of all. And we've got parents with blood type A and B. So let's have the one parent up here. Let's go with blood type A. So they've got blood type A and they need another allele. Okay, we'll come back to that one in a minute. Let's just put a little question mark. And we've got the second one, which is blood type B. So they must have that one there. And again, we need that second allele there. Right. So we're going to do our Punnett Square as per usual, but I'm just focusing on the fact that we're going to end up with 4 different blood types. Now, if you think about what the blood types are, you can have blood type A, you can have blood type B, you can have blood type O, and the fourth one is AB. We need to get all 4 of these. O is recessive. That means that if I'm going to get O, I need to have an O from each parent. So that has to be an O there, that has to be an O there. And now, this is blood type B from one parent, blood type A from another parent. I should be able to get all 4 of these done. Let's have a look if it works. Right, codominant alleles. So that's going to give me blood type AB. So tick, we've got that one done. That's AO, so that's going to give me blood type A because O is recessive. And likewise, that one's going to give me blood type B. I then have 2 O's combining together. That's going to give me blood type O. So using that combination, we have managed to work out all 4 possible blood types. Just one quick word about this codominance thing before we move on. I've used the notation that we're very used to, where we've just got the alleles written out. Sometimes questions will do it in a different way, and you don't need to get alarmed by it. The principle is exactly the same, but I'm going to take this example here and show you a different way of notating it. So sometimes you will have the allele given like this. So this is using the blood type example. With the protein we're talking about is actually called an immunoglobulin, and sometimes it'll be written like this. So these are that's a person with blood type A, for example, just like we had in our example over there. The only difference is that we use this capital letter here just to show it's an immunoglobulin, and the actual allele type is written as a little superscript up there. The principle is exactly the same. You're talking about one allele, or in this case actually it's 2 alleles, so different alleles of one gene. So this is, for example, blood type B, this would be blood type AB, and this would be blood type O. Sometimes you can see it, for example, with the colour example, you might see that written like this, so colour red, colour white, together they give pink. They use this kind of notation to illustrate that there is codominance, and rather than one being dominant to the other. Don't panic if you see it, the principle is exactly the same. The crucial bit here is this is telling you what form the allele is in. Okay, so different annotation, same result.